Assalamualaikum and hi everyone. In this video, my group will explain about SDS page and its process. These are my group members, which are Nisa, Asyiqin, Sharifah, Fatimatul, Aqilah and Nur Haziqah. These are the list of contents that will be discussed in this video. Firstly, we need to know what SDS page is. SDS page is a sodium dioxide sulfate polyacrylamide gel electrophoresis. The importance of SDS page is used to obtain high resolution separation of complex mixtures of proteins. Next, SDS page act as a detergent to break the disulfide bonds of protein disrupting tertiary structure of proteins. The principle of SDS page is a charged molecule will migrate to the electrode with the opposite sign when exposed to electrical field. The small molecule will migrate faster than large molecule due to less resistance. The structure and charge of proteins also can be the factors of the migration of molecule. Discontinuous SDS page is the common method used for protein, while continuous SDS page is for nucleic acids. These are the list of materials and apparatus that will be used in the preparation of SDS page.
Now let's prepare the resolving gel in the beaker. Ensure all the solution have the same temperature. Add 1.5 molar of thrice hydrochloric buffer with the pH of 8.8 to a beaker containing distilled water. The pH ensure the mobility of the negatively charged polypeptide change reaction towards the anode. Add 4 ml of 30% of acrylamide to the beaker. Acrylamide is a monomer which will be polymerized, and the build in this acrylamide ensure the cross-link to matrix a fairly well-defined structure of the polymer. Add 10% SDS solution into the beaker and left it for 10 minutes. This allowed the air bubble to liberate for a while. This is important as the polymerase is carried out by the change radical mechanism. Add ammonium persulfate and tetramethyl ethylene dynamite timid at the end. Timid catalyzes the polymerization of acrylamide. If the timid is being added too early, the gel will set in the beaker rather than between the plates. Pour the gel in between the plates up until the gel is at the same level as the mark shown. The presence of the air bubble can be fixed by adding a layer of isopropanol on the top of the gel. Wait until the gel has polymerized. Dry up the isopropanol using the filter paper or just pour it off.
Mix 1.35 ml double distilled water, 0.235 ml acrylamide stock, 0.25 ml upper stock and 20 microliters of 10% SDS. Drain the isopropanol, rinse the top of the gel with DD water and dry with clean wipes. Add 20 microliters of 10% APS and 2.0 microliters of TMAT into the stacking gel solution. Mix well. Pipette 1 ml stacking gel solution on top of the resolving gel in between the plates. Fill to the top and insert the comb into the solution. After the gel is formed, unclip the casting frame with the gels from the casting stand. Remove the glass plates from the casting frame. For SDS page, label the microfuge tubes corresponding to your samples. Pipette 40 microliters of the fish extract and add 40 microliters of the sample buffer. Tightly secure the gel with the short plate facing inwards onto the running module. Each running module can hold 2 gels. If only one gel is being run, you need to use the buffer dam in place of the second gel. Insert the running module into the tank. Check if the lid fits properly. Pour the 10 times dilute running buffer solution into the running module until wells on the 2 plates are fully submerged. Check the running modules aren't leaking. The tank only need to be filled halfway with the buffer solution if running 2 gels, and ready to load the samples. Install the loading guide on the gel to make it easier to load your samples properly into each lane. Carefully load 10 microliters of sample solution into each well. The pipette tip should be inserted into the well in between the plates, deep enough to prevent overflow of the sample to the neighboring lanes. Slowly release the sample solution. Remove the pipette tip, change the tip, then load another sample. For the molecular weight marker, you also need to load 10 microliters into the well, and it should be loaded in one of the center lanes for easy comparison.
Firstly, when all the samples are loaded, close the lid on the tank and insert the electrodes properly into the power supply. red wire into the red slot and black wire into the black slot. Set the power to 20 mA per plate and run for 1 hour. The power can also be set to voltage. The voltage used does not depend on the number of plates. You should initially observe the sample or standard solution to be condensed into a thin band through the stacking gel. Then, the band will gradually migrate down the resolving gel. When the blue dyes have reached close to the bottom of the gel, stop the run, unplug the power supply, then remove the running module from the tank and drain the running buffer. Remove the glass plates carefully from the running module, then carefully use the gel releaser to remove the gel from the glass plate. You can trim off the stacking gel carefully before proceeding to staining. Transfer the gel into a square petri dish, then pour the Commercy Blue staining solution into the petri dish, submerging the gel fully. Place the square petri dish in a metal tray to avoid spillage in the incubator. Then, place in a 15 degrees Celsius incubator for 30 minutes. Remove the gel with a tweezer and make sure you can observe clear bands on the gel before discarding the staining solution and replacing it with the destaining solution. Add a piece of clean wipe or absorbent paper towel to help absorb the dye. You may also want to change the destaining solution every 15 minutes until the background of the gel is transparent. When the destaining is complete, store the gel in DD water until you can scan and analyze it with the densitometer.
Western blot procedure is typically cell and molecular biology procedure widely used in protein analysis. Also known as immunoblotting, the methods allow researchers to determine presence, size, or quantity of particular protein in a sample. The principle of Western blot is based on polypropylene gel electrophoresis and antibody. Western blotting technique steps are including the sample preparation, gel electrophoresis, protein transfer, blocking, primary antibody incubation, secondary antibody protein detection, and analysis.
There are 7 steps in Western blotting. First step is sample preparation. It involves the extraction of protein from different samples such as cells. They broke down by sonification, and once done, quantity protein needs to be determined. Second step is gel electrophoresis. Separation protein based on their charge and molecular weight. We will use native page. Third step is protein transfer. Once gel electrophoresis procedure is done, they are transferred to solid support membrane, such nitrocellulose. An electric current as electroblotting technique is applied to protein gel sandwich between the membrane and the filter papers. We perform this on wet transfer. Fourth step is blocking. Blocking is a crucial step in Western blotting technique. Blotting membrane have a high affinity for protein. Blocking binding site after transfer to avoid further non-specific binding of the assay detection antibody. It will reduce the noise in Western blots, result to more precise findings. After blocking, antibody probing is performed. The 5th step is primary antibody incubation. At this stage, the primary antibody is used to probe the membrane and binds to the prey protein, depending the antigen primary antibody selected.
The 6th step are secondary antibody incubation. Wash the membrane using an antibody buffer solution to reduce background and remove excess antibody. After that, second antibody incubates the membrane. The secondary antibody will bind to first antibody that has internate with prey protein and is detectable. The last step is protein detection and analysis. Protein detection is achieved when conjugated enzyme interact with the substrate introduced to whole membrane, causing chemical change in the form of a colored substance, allowing the researcher to mark the location of the prey protein. So, in conclusion, application of Western blotting can help in determine the diagnosis of infectious disease, determine in a protein is expressed, and many more.